All right, everyone. Natalie is having some issues connecting, but she should be here soon. And that will be everyone who's presenting today. So thank you everyone for coming to attend. This is the 2020 virtual REU Eco-Evo Symposium, and we have a great schedule lined up for you. How this is going to work is I'm going to share my screen. I'm going to give a little bit of an introduction to the whole program, and then I'm going to pass the mic off to our students who are then going to be able to remote control the presentation and click through on their own. So hopefully everything works. Fingers crossed. Okay, can everyone see my screen? All right, thumbs up from Callie. All right, so this is the symposium that is ending our 10 weeks of research in the Ecology and Evolution virtual REU group. And all I want to do today is give a very brief background on how this came about and what we did this summer, and then let you hear all of these excellent talks that our students have prepared. So I've been involved in mentoring undergraduates for a while, and all sorts of wild programs, from working on the green roofs in New York City to collecting ticks in the forests of Westchester. And I've really loved working with a diverse group of students and seeing how they change over time and benefit from the REU program. And what the challenge, of course, of this summer is that there are no in-person programs. So what I wanted to do was create something that could be maybe a facsimile of an in-person program and replicate some of the best factors that make them so useful. So what I wanted to do was really provide some framework for the students to learn from each other and to learn from mentors from a wide array of backgrounds. So how do we do that virtually? So these are my excellent mentor companions: Callie Chappell, Sarah Wuchik, Patrick Clay, Melissa Burt, and Kathy Toll. And they have been outstanding this summer in helping me put this on and really provide a REU program for these students. And what we were able to do was basically work with each of our backgrounds and strengths and provide different opportunities to the students in terms of teaching them things that can help in future scientific careers, like reading scientific papers, writing scientific papers. We got a lot into coding. That was a challenge, but I think a fun challenge. And learning some things about the sort of unwritten rules of science in terms of how do scientific ethics work, and how can you manage your own interactions with other scientists. So that has been really exciting, and I thank these other mentors for all of their hard work that they didn't need to do. So I really appreciate it. And then, of course, a big advantage of the virtual setup is we were able to bring people from all around the country, and even from other countries like Canada. So we had a career panel where we brought in people from many different backgrounds, and they talked to the students about careers in science. And we got help from Dr. Megan Duffy and Abir Siddiqui, and they talked to the students about their specialties. Sorry, I have a train going by behind me. So amid the challenges of the virtual program, there were some advantages. We could bring in a wide array of different specialties, and we could allow students to come together that would not normally have been able to. And with that, I would like to introduce our students. They come from a wide, I keep saying wide array, but a diverse group of institutions. And this is how they looked for most of the summer. So that's what I get to show you. But you will greatly enjoy their presentations, and I hope you stick around for all of them. First up today is Gabby Dugan of Virginia Tech. So I'm going to pass remote control to her.
Okay, can everyone hear me?
Yes.
All right.
Would you like to turn on your video?
Yeah, sure. Okay. So, hi everyone. My name is Gabby Dugan, and this summer I worked with Melissa Burt, and we studied the response of ant communities to habitat connectivity and time since prescribed fire. First and foremost, I just want to say thank you to all of these organizations, and especially to Melissa for making this REU still happen, even virtually and for the most part over Zoom. Sorry about that. Acer, it's having some trouble moving forward. Okay, so the questions that we're going to be presenting is how are ant communities affected by habitat connectivity and time since prescribed fire, as well as does nest site preference predict ant species response to habitat connectivity and time since prescribed fire? So firstly, habitat fragmentation is when you divide habitats or landscapes into smaller, more isolated pieces. And as you can see in this figure, it decreases habitat connectivity and increases the amount of edge that a habitat has. Why it matters is it creates a loss of biodiversity, and it's rapidly increasing because of human development. And one way to mitigate habitat fragmentation is through the use of corridors. So as you can see in this image, the purpose of a corridor is to increase habitat connectivity. And these are a couple images of some man-made corridors, which again aim to connect habitats. The Savannah River Site Corridor Experiment is where our study was located. The purpose of it is to test the effects of habitat fragmentation, specifically habitat connectivity and edge effects. In the corridor, in the experiment, there are 7 blocks with these 3 patches. The winged patch has low connectivity and high edge, whereas the connected patch has high connectivity and high edge. And between these 2 patches, we test connectivity effects. Whereas the rectangular patch has low connectivity and low edge. So between the rectangular and the winged patches, we want to test edge effects. For the data collection, these were not data sets that I personally collected, but it was ant community data sets that we used from the corridor. They were collected annually since 2015's prescribed fires in all 7 blocks. And in each of the patch types, there were 22 pitfall traps. So our study, again, it was located at the Savannah River site. This is a picture of one of the blocks. The patches are clear cuts in a longleaf pine plantation, and the site undergoes prescribed burns to maintain the habitat. Why we study ants here is that they're indicators of biodiversity and overall ecosystem health. And in our system, we have almost 50 different ant species, which makes it possible to test things like species richness, biodiversity, as well as functional traits. And in this specific project, the functional trait we focused on was nest site. How I collected the data was through ant databases and literature mining for all of the almost 50 species we have in our system. And for the data analysis, I counted up the species that nest in soil, litter, and wood. So going back to our questions, the first one we asked again is how are ant communities affected by habitat connectivity and time since prescribed fire? This is what our figure looks like. It's data from 2015, 16, and 17, and it's going to be color coded according to the patch type. In addition, I used R to count up the average number of species collected per trap within each one of these 3 patch types. So this was the figure for the species richness in the patch types. And the first thing we noticed was that there was a significant effect of time. We saw that species richness was increasing over time after the prescribed burn. We also saw that type had a significant effect. As you can see in this figure and through the p-values, the winged patches have higher species richness than the connected patches. One of the reasons we think this could be is that the connected patches have been shown to have more fire ants, and we think that this could be causing the decreased species richness in the connected patches versus the winged patches. And the second question we looked at was, does nest site preference predict ant species response to habitat connectivity and time since prescribed fire? Again, our figure is data from 2015, 16, and 17, and it's going to be color-coded according to the patch type. The first one that we looked at was soil nesting species, and what we found was that there was not a significant effect of time or patch type on the species richness. We think that this could be because the amount of soil available for nesting didn't change substantially based on the patch type or the year. The litter nesting species, as you can see from this figure, there are far fewer litter nesting species than soil nesting species in our system. However, when we looked at the pattern, we found that year had a significant effect after the fire. And the reason for this, we believe, is because after the fire, there's not as much litter on the ground of the corridor experiment. However, throughout the years, it starts to accumulate so that the litter nesting species, there are more of them over the years. For the wood nesting species, we saw a really similar pattern where year had a significant effect, but patch type did not. And again, the reason for this, we believe, is that after the fire, the woody debris that ants are nesting in starts to accumulate, so there can be more wood nesting species in the experiment. These were the conclusions from the project this summer, and I will keep these up and ask for any questions.
Okay, it doesn't seem like I can keep this full screen and manipulate things at the same time. So if anyone has any questions, you can ask them on the Zoom or put them in the chat on either the Zoom or the YouTube live stream.
I have a question, Gabby. Great talk, really good job. My question is why you chose to focus on ants and how you feel like ants might be the things that you see with ants might be interactions with other species.
So that's a great question. And so one of the reasons just in our system why we chose ants is because there are so many different species, which allows us to look at things, look at different functional traits and compare different species response to disturbance. But also ants are prevalent not just in our system, but across the world. So it's really important to see how they're responding to disturbances since they are so widespread. And then it's been shown that ants are indicators of overall ecosystem health. So because we used ants as a system to study overall biodiversity, and ants and plants can have similar reactions to disturbances. So we also wanted to study that. And moving forward, we hope to compare the amount of vegetation in the corridor with the ant species communities to see if there are similar effects.
Very cool, thank you.
Nate had a question in the chat, and this is how specialized or generalized are the ant species in terms of habitat preference?
That's a really good question. So that was another thing that we looked at this summer. We have a lot more specialists in the system, which is pretty interesting because you would expect there to be more generalists, but because our system is adapted with fire, a lot of the species that we have are soil nesting, and they're soil nesting specialists. However, when we first did our literature search, what we did was we divided them into 6 categories to see the different crossover with habitats, but we saw that we had a lot more soil specialists than any other category or functional group.
Great. Thanks so much, Gabby. We're gonna move on to our next presentation. This is from Lexi Golden, who is in Callie's lab. And unfortunately, she could not make it to the Zoom call, but she has pre-recorded her talk, so we can enjoy it through that. So I'm gonna go full screen here, and it should play right away. Is it still silent for everyone? Sorry about that. Can anyone hear this video?
Golden, and I had the opportunity this summer to continue working with the Fukami Lab and Callie Chappelle on our Nectar Micro project. Today, I will be explaining the experiment, some of our findings, and why it is important. Here's a quick overview of what I will be covering today. First, I will be discussing some background information, then describing the experiment and the analysis that I have been working on this summer, and finally, the importance of our research. We have evidence that rapid evolution can affect ecological processes. One of the most popular examples of this is Darwin's finches, which were finches that possess different beaks based on their diet. And this leads us to our question, which is how do evolutionary processes affect metacommunities? Here is a diagram that helps in understanding what a metacommunity is. Within a local environment, which we call a patch, there are interactions occurring between organisms. However, there are times where organisms may migrate to other patches and begin interacting with organisms that live in that community. All of these patches that are connected by dispersal vectors are a part of a larger metacommunity. There are 2 possibilities for how evolutionary processes affect metacommunities. First, we have evolution, which would lead us to believe that it is solely genetically based. Secondly, the other possibility is ecological processes, such as priority effects. Priority effects are the impact that a particular species can have on a community development due to the order of arrival. Or we could have a mixture of both, which is most likely the case. Now we can begin to discuss the specific system that we are currently studying. We are working with the nectar yeast M. rickelfii, which is found within the sticky monkey flower, native to Northern California. Returning back to our diagram on metacommunities, you can see how this plays a role within our study system, where each flower represents a patch, and the organisms living within the flower nectar are connected by pollinators. This creates a larger meta-community. There are two organisms that interact within the nectar, yeast and bacteria, and they are constantly in competition with each other. So to reiterate our question, can yeast rapidly evolve to compete better against bacteria? In order to answer our question, an experiment was designed that started out by isolating yeast from the nectar of a wild sticky monkey flower. Once the yeast was isolated, it was then put through a series of transfers to simulate generations of evolution, similar to the evolution that would happen when yeast moves from flower to flower in the field. We wanted to see if the yeast would evolve to compete better against bacteria, so we used a special nectar that we made in the lab. To make this nectar, we grew bacteria in the nectar and then filtered it out before adding the yeast. For purposes of ease, we refer to the isolated yeast as ancestral yeast to distinctly differentiate it from the evolved yeast. For the experimental evolution, we transferred 1/10 of the yeast suspended in normal synthetic nectar, which was 10 microliters every 2 days, to fresh nectar. This created a bottleneck effect, simulating the pollinator's process, where pollinators move a small portion of microbes from flower to flower. This was then completed over the course of 2 months, or 30 transfers, which represented the length of the flowering season for the sticky monkey flower. Since we know that yeast and bacteria are constantly competing in nectar, this leads us to our question: which yeast competes better against bacteria, ancestral or evolved? Luckily, we already know the answer from lab experiments I and other undergrads completed earlier this year. According to our results, evolved yeasts are less affected by bacteria. Looking at our graph, the y-axis demonstrates final yeast density and the x-axis represents the two types of yeast. The more affected that the yeast is by the bacteria, the lower the points on the graph would be, and vice versa for the less affected yeast. But what is the basis behind this? Why does the evolved yeast compete better against bacteria than the ancestral yeast? This leads us to our next question: which genes are most sensitive to mutations in nectar yeast? To answer this question, which is what I've been working on this summer, I ran a series of analytics on the sequencing data. This started out by extracting the DNA from the yeast, which was completed in the lab. The samples were then sent to the Stanford Functional Genomics facility for sequencing. Once that was completed, we began working with the files using various programs on the command line to better understand the sequences. Here is an overview of the programs that I used and will be using to complete the analysis. I explain what each of these steps do now. First, I ran the FastQC to quality check the sequences, which ensure the data is of high enough quality for the next step. Once I ran the FastQC, I was able to move on to the BWA-MEM portion of the analysis, which reads the sequences of the yeast and aligns them to the reference genome provided to us by prior researcher Dr. Manpreet Dhami. Shown here is this illustration. The reads match up to the corresponding base pairs on the reference genome, given that there is a high enough reading depth to cover the entire genome. Once the entire genome is covered, the best reads are then strung together to produce the alignment results. Next, the ancestral genome will then be compared to the evolved genome using FreeBase, which is a program that will locate SNPs or places where the gene sequence differs by one base pair. As seen here, the sequences are identical for both the ancestral and the evolved reads, besides the A and T at the same position. Once this process is complete, we will take the genes that contain SNPs and blast them, which will tell us what the closest known gene sequence is and give us an insight into what the function could be. As seen here, the gene that contained the SNP would have certain functionality, for example, a binding protein gene. Since this portion of the analysis has not been completed yet, here is a sample of how we might display our data. This particular figure, which is unpublished data from a different experiment in 2018, is a representation of what our data may look like. For example, the y-axis would contain different gene functions, such as transcription and metabolism, and the x-axis would be the number of variants or mutations in genes of that function. This would mean that the longer the bar, the more variants there are in the genes of that functionality. So overall, we addressed two major questions. Part 1 is which yeast competes better against bacteria, ancestral or evolved. We now know that the evolved yeast does significantly better than the ancestral yeast. We don't have the answer to part 2 yet, as I'm still working on those results. It is still important to be interested in ecological processes because they are happening all around you, whether you know it or not. Bacteria-dominated flowers can actually change the conditions of the flower's nectar, which can harm the plant and also make it less likely to become pollinated. Yeast, on the other hand, positively affects the flowers, making it more attractive to pollination. However, not only does this affect the flowers, but it can also affect the foods and drinks that you eat. For example, kombucha is made using a mixture of yeast and/or bacteria. Yeast can make a sweet kombucha, while bacteria will make a bitter, salty kombucha. Beyond this system, this research shows that evolution can occur very rapidly, right under our noses in the lab and the field. We have shown that this evolution can influence how all species compete, and my ongoing work will show in our system which genes are important for determining the outcome of competition. Thank you for listening to my presentation today. I would like to conclude by thanking my mentor, Callie Chappelle, along with all of the members of the Fukami Lab, as well as the undergraduates on Team Nectar Microbe.
Okay. Well, unfortunately, Lexi won't be able to answer any questions, but if you would like to leave them in the chat, I think that she could chime in the future. Next up.
I'm sorry, sir. I just wanted to say briefly, if anyone has questions, you can send them to me or you can send them to Lexi directly, and I'm happy to send those to Lexi and she can get back to you asynchronously.
Perfect. Thanks, Callie. So next up, we have Natalia Hernandez, and she will be presenting a poster presentation on her research in Daphnia. Are you ready, Natalie?
Yes, I'm ready.
Perfect.
Okay, hi everyone. So just to give you like an overview of what Daphnia are, they're plankton-like crustaceans. They're very important in lake ecosystems, and they're used as a model in understanding disease dynamics because they serve as a parasite host system. And also just to explain how I approach this poster, I wanted to give an intersectional review. During this whole process, we read a bunch of research papers, and a lot of the review papers are very effective in giving us a good understanding of like different of the material that we were learning about. So I wanted to create a poster that emphasized different aspects of this research that I learned throughout this program. So just to begin, if you look up at the diet section, I wanted to demonstrate that the Daphnia's main source of diet is algae, in particular algae that's towards the surface. There was research that was conducted that demonstrated that algae from shallower lakes rather than deeper lakes were a better source of nutrition for Daphnia. So similar to like how humans want to eat the most nutritional foods, Daphnia, in their case, it's eating the algae that's closest to the surface. It gets the most sunlight, and this higher quality diet leads to better health for Daphnia. Just like us, they need a good diet to support their metabolism and their growth. Also, when Daphnia are infected with parasites, there's a competition for the resources that are gained from their diets. And a lot of times, the different things that they eat, whether it's algae or other types of, like for example, cyanobacteria and phytoplankton, some of it can be more beneficial to the parasite rather than Daphnia. In this case, if you look at my little pie chart next to some of the algae, glucose is the most beneficial to the Daphnia because iron, carbs, and protein actually increase virulence. And to sort of segue into the next section, which is behavior, the Daphnia will behave in certain ways to access nutritional foods, but also to avoid predators. Also, there's a genetic component called phototaxis, where some Daphnia are more likely to go up to the surface rather than to, I guess, towards the sunlight. rather than going down to like deeper depths. So this is actually called diel vertical migration. And basically what happens is if you follow the little blue path all the way on the right side, the Daphnia will migrate all the way up to the surface during the nighttime when they're less likely to be engaged with by a predator. A lot of times these tend to be fish, and if you look at my big fish that's already eaten a couple of Daphnia that have been infected, they come out and they're able to see the Daphnia more so during the sunlight. The Daphnia have a very clear carapace, and so they can be seen more easily when there is sunlight. So they will migrate to lower depths during the day. However, at nighttime, when the phytoplankton, a lot of times it can be fluorescent, they'll go up to the surface and they're getting the most sunlight. So then that's why they migrate during the nighttime. Also, something that is influencing Daphnia and how their movement is if they're infected. So some research conducted by one of our lab members demonstrated that being infected by a fungus called Mechnicalia, I think it's Mechnicalia, also known as Mech, will influence the swimming speed of Daphnia. Similarly, the other types of parasites that Daphnia can be infected with is also Pasteurella, and which is a bacterium. And something that is also key to know is that in the center of my poster, to sort of emphasize the intersectionality of the study of Daphnia and how these different areas of study involve one another. If you look, you can see that, for example, for diet, there's a main emphasis on the resources. We talked about a high quality of resource, and then quality. And then we also talked about competition, disease, and how having a higher quality of diet can make them less susceptible, and it could also be preventative. And in regards to behavior, that also helps with prevention, susceptibility, and also in regards to population density, depending on number of Daphnia, and as well as disease. Something that is also important to consider is that there's fish pheromones that are the chemical cues that allow the Daphnia to know that there is a predator in their presence, and this is what also helps them in regards to their behavior. Also, the Daphnia have certain features such as antennae that help them move in through the water. They tend to twitch or they'll swim using their antennae to sort of direct them. And they also have appendages towards the lower part of their body, which is what they use to filter feed and also to release their babies during reproduction. Something that is also notable about the Daphnia behavior is that they can produce sexually or asexually, and this is also dependent on the environment and if it's favorable. And to sort of transition a little more into our next sphere of research. So in regards to all these interactions between Daphnia and their environment, their predators, and even other Daphnia, there's importance to emphasize how this can sort of serve as a mechanistic process. There's a lot of different factors involved. So when talking about multi-parasite ecology, and Daphnia being infected by one or more parasites, it influences the Daphnia in many ways. So if we're looking at my first little plant on the left with the two fishes with host resources in the immune system, if you start from the bottom, you'll see that this is, sorry, I was timing myself to make sure I didn't go over, so I'll speed up a little bit. But so as you see on the left, there's the first trophic level that is important is in regards to host resources. This could be where in the Daphnia it's infected. It can be their gut. It can be in their blood, whereas actually how the pathogens spread. And then also not only that, but also the parasites that infect it. And that's the second trophic level, the parasites and their parasite guilds. So that also goes into competition, their parasite host dynamics, and the population density is also important. And then the third trophic level would be the immune system, because different pathogens will influence the immune system in the sense that certain pathogens will create a cell-mediated response or humoral response. And this will have different effects on the Daphnia because the presence of one parasite may make the immune system be more preventative against that parasite, but there's also a presence of another parasite, it may not be as well guarded. And then sort of to transition into multi-host ecology, similarly, there's also a mechanistic process with having to consider many different factors. For example, multi-host ecology consists of a lot of community disease dynamics, and something that's typically understood is that this can be determined by host competence, but actually population density is important to consider as well. And that's why different models have been made, mathematical models and mesocosms, or mesocosms, which are used to sort of model this natural environment and to also gather more information, and both for use. And so to further continue, basically in this mesocosm, there's the parasites, and then in this specific research, invasive hosts and native hosts. And invasive hosts tend to have higher competence while the native hosts have lower competence. So the invasive hosts are very good at spreading disease, and the native hosts have an amplification effect on the invasive hosts, while the invasive hosts tend to dilute disease in the native hosts. So the way that this works is that when you think about the population composition, and this is all demonstrated on that little rock for multi-host ecology, sorry about that. If you look into the light blue circle, so basically the population composition, there's competition, intra and interspecific, between the different types of hosts. So when you add native hosts to invasive hosts, what will happen is that there will be an increase in the total host density. This overrules the average host competence. So a way to think about this is if you have a population of people and no one's lactose intolerant, and then you add someone or a couple people that are lactose intolerant, it will increase the average lactose intolerance. So this is similar in that sense. And as a result, there's an increase in the infected host density of invasive hosts, and this leads to the amplification of the disease. Meanwhile, if you were to add invasive hosts to the native hosts, it will decrease the total host density, and this would also overrule the average host competence, and then there would be a decrease of the infected host density of the native hosts, which leads to dilution. But overall, as you can see, there is a very intersectional overview of all this type of research, and I thought it was important to demonstrate how they are all overlapping. And I would like to acknowledge before I finish the Duffy Lab, and with special regard to all my mentors. Thank you.
Great job, Natalie. And I should have mentioned at the start that Natalie was in the Northeast and lost power this weekend, so she has pulled this off at the last minute. So great job with that. I think we have time for one question. Let's see, check on the chat.
I was curious, Natalie. So you said at the beginning you were talking about diet. You were saying that, you know, if some food is better for host, some food is better for parasite, and the host, you know, if it has a lot of glucose, that's better for the host. If it has a lot of iron and other stuff, that's better for parasites. Do you know, is that specifically for Daphnia, or are there does that reflect a broader trend for food as like across many animals with their diets? Is it typically better that high glucose diets are better for hosts, or do you have any knowledge about that?
I think this was from a paper. This was definitely, well, it was from a paper, but I'm unsure. Like, I don't exactly remember, but I think it might have been more general. But to crustaceans like Daphnia, and just like in particular to microparasites. So I don't know if it's specifically to Daphnia and specifically type of parasites that that infect them, but it's not as broad. It's a little more specific to like, I guess, organisms that are similar to them.
Thank you.
Awesome. Great job, Natalie. Next up, we have Sydney, who was mentored by myself, and Kathy Toll. I'm going to give you control, Sydney. Start away.
Sweet. All right, so hi, like you said, I'm Sydney, and I go to MS. So this summer, I studied how the perennial plant switchgrass has changed over space and time. Hold on. Is it letting me? Oh, okay. Too far. Sorry. Okay. So how it has changed over space and time. The importance of this study boils down to maximizing ethanol yields. Due to its hardiness, carbon requirements, high productivity, and other factors, switchgrass is being considered to be a biofuel crop. So it is essential to learn all that we can about it. Temperature has a strong link to growth rate of switchgrass and thus its ethanol yields. And with climate changing so dramatically, as we all know, studying its effects on switchgrass over space and time is vital. There are two known ecotypes of switchgrass that have locally adapted to their environments. Upland plants can survive the harsh winters of higher latitudes, but are small because of their short growing seasons. While lowland plants are large and can have multiple growing cycles per year, and they also have higher pathogen resistance. Genetic studies suggest that 3 populations exist within these ecotypes that have also locally adapted to their environments. They're known as the Midwestern, the Gulf, and the Atlantic populations. All of these can succumb to pathogens that can cause an ethanol loss of up to 50%. So with all these facts in mind, these are what inspired my research for this summer. So my first objective was to identify how latitude impacts the phenology and infection rate of switchgrass. And I hypothesized that at higher latitudes, infection rates would decrease, and that the 3 populations of switchgrass would have different phenology latitude relationships. My second objective was to determine how the phenology of switchgrass has changed over time. And I hypothesized that as temperatures have increased, switchgrass has begun earlier growth cycles in recent years. My third objective was to analyze the impacts of biotic and abiotic factors on the phenology of switchgrass. And I hypothesized that biotic factors such as pathogens will cause delay in growth cycles in the southern plants and earlier growth in the northern plants. And then my last hypothesis was that abiotic factors, increased shade and water availability, would cause delay in growth cycles. Okay. So iNaturalist and Herbaria databases allowed me to gather from all over the United States and from all different time periods that I would not have been able to do on my own. The main data points I collected to satisfy my objectives were pathogen presence, latitude, panicle angle, collection date, and habitat information. The observation, as seen here in figure 2, that panicle angle increases as the plant develops, allowed me to correct collection date to an estimated flowering date in order to standardize my samples. Figure 1 down in the left demonstrates how I collected panicle angle and what I looked for in pathogens. The easiest one for me to spot was known as bipolaris or leaf spot, and that's what's shown here. This figure emphasizes the power of iNaturalist by showing all of the samples I was able to collect. Also, the colored circles show how I estimated population groups because I did not have access to their genomes. Now for what we've all been waiting for, the results. Due to time constraints, I will focus on my most exciting finds today. So to answer my first hypothesis, I found that latitude has a significant relationship with pathogen frequency, as seen here in figure 4. More specifically, as latitude increases, pathogen frequency decreases, which is interesting when it is known that the more southern plants have higher pathogen resistance. This is possibly because there are longer growing seasons, so there's more chance for it to become infected, or maybe that more pathogens exist in the south because there's no harsh winter to kill them off. My second objective also saw promising results in that I found a significant relationship between year and day of year of flowering. The Atlantic and Gulf populations have been flowering later as the years progress, while the Midwest has been flowering earlier. This most likely is due to how temperature affects the plant's growth rates. I would speculate that an increased temperature delays growth in the lowland or in the southern plants, which is opposite of what other studies have found, and that the Midwest start to grow earlier because the higher temperatures have told them to start growing so that they can take advantage of their short growing seasons. My hypothesis 1B, 3A, and 3B, I did not find any significant support for, but hypothesis 1B, which I was hypothesizing that 3 different populations would have different phenology latitude relationships, I did find marginal support that the slopes between the 3 lines were different. All right, so what is the takeaway from my whole summer? First, as latitude increases, the presence of pathogens decreases. This is really important to note, mostly knowing, as I said in the beginning of the presentation, that the presence of pathogens can decrease ethanol yields by up to 50%. So if we're wanting to use this as a biofuel crop, that's not really a good thing. Second, over time, the flowering day has been changing. In fact, I took a quick average, and in about the past 100 years, there's been a flowering day increase of about 21 days. Again, also something significant, because if it's flowering later in the year, it's not getting to take full advantage of its growing season. So we're having smaller plants, therefore less ethanol. And then finally, an overarching key point from this whole summer was that online databases can produce results, which is especially important during these COVID times when a lot of us aren't able to do science like we normally do science. So in the future, if someone were to repeat my experiment, I would definitely increase the amount of samples. I only was able to collect a fraction of the amount of samples because I was only one person and, only so much time, but there were so much more available. Also, I would have an expert analyze samples to ensure the pathogens on the plant were correct. I was not an expert, so that's why the bipolaris was easiest for me to spot because it was pretty obvious, but I'm sure there were some that I did miss. Also, I would find a better reference data for the panicle angle. This is just to get a more accurate estimated flower state. And then in the future, I would love to see someone continue my experiments using common garden experiments in order to support the results that I've seen in these virtual observations. And then finally, I just wanted to do a quick acknowledgement to my mentors. They have been amazing, and I have had such a great summer and learned so much. It has been such a beneficial experience to me. And also for the National Science Foundation and the University of Texas for funding my project, and to Dr. David Lowery for providing me with this opportunity. It has meant so much to me. So, yeah.
Thank you, Sydney. That was great. Any questions for Sydney?
I have a question. This seemed like a really great use of iNaturalist data. Do you think it could be used to look at other interactions too, like maybe herbivory?
Because in the beginning, I was going to do like interactions between species. Like is that kind of what you mean, like interaction between species?
Yeah, like would you be able to measure rates of herbivory? So like plant, insect herbivore, plant, like mammalian herbivore interactions with these kind of data? Could you tell with the pictures if there was herbivory on those?
I would say, because at the beginning I was going to do some sort of interaction, but it was kind of hard. Because they only really identify the main plant, so like the switchgrass, they don't really identify anything around it. And sometimes the picture just isn't good enough quality to like identify. So I don't know if it would be good in that, but in studies of just the plant that it is called, I think they even do insects on there. I'm not 100% sure about that one, but those kinds of studies I think would work, but not maybe not interactions.
Nate had a question in the chat. Sydney, do you have any advice for anyone interested in using iNaturalist for data collection?
Ooh, let's see. Definitely gonna have to have patience because iNaturalist has a lot of pictures and not a lot of people know how to take quality pictures. So that is one thing that I really learned is you have to be able to have patience to search through all of them. And yeah, I guess that would be my main piece of advice for going through iNaturalist is the patience.
That sounds very important. All right. Thank you very much, Sydney. That was excellent. Next up, we have Saye. Oh, we have one more in the chat. Okay. All right. Are you ready, Saye?
Yeah, sounds good.
All right. I'm going to stop my video.
My name is Sai Regani, and I'm a sophomore at Stanford University. And this summer, I had the opportunity to work with my mentor, Priscilla, and our principal investigator, Professor Fukami, to study the diversity and distribution of nectar microbes in New Zealand plants. Now, in this presentation, I'm going to go over the objective of our study, briefly go over the methods we used, and then explain 2 of the measurements we used to understand the biogeography of nectar microbes in New Zealand. And those will be alpha diversity and beta diversity. I'm just trying to go to the next slide. Okay, and so why nectar microbes? The communities of fungi and bacteria that are found in nectar can increase plant pollinator interactions, influence plant fitness, and provide nutritional value for pollinators. And this study is the first survey of nectar microbiomes in New Zealand, so it can provide us greater insight into those plant pollinator systems. And the methods that we used for this study were sampling the fields in both the North Island and South Island of New Zealand, extracting and amplifying the DNA using PCR. And then in order to identify the different bacteria phyla, we barcoded the 16S RNA strands, and we put all of that information into the computer and used bioinformatics to analyze our data. And so our principal question was, how do geographic location and plant specificity affect the community composition of nectar microbes? And as you can see from the map to the right, these plants were sampled in both the North and South Islands, and for the most part were only sampled in one of them. However, the flax and manuka plant were sampled in both of the islands. And some different hypotheses we have for why there might be differences in these communities is because some of these plants have different plant morphologies. For example, as you can see, the flax plant has more of a tubular characteristic, whereas the manuka plant is more flat, and that can affect the accessibility of different pollinators and therefore the nectar microbes. Another reason could be that there's temperature variance between the North and South Island. In addition, there might be pollinator variance in different regions. And so the first thing we're going to look at is the alpha diversity, which is a measurement of the richness and evenness of a microbial community. And so in this graph, we have the plant species on the x-axis, and we have Shannon diversity, which is a way of measuring alpha diversity on the y-axis. And the higher our Shannon index is, that means the greater richness and evenness we see in that community. And up above, you can see the pairwise ANOVA post hoc that we did. And those represent if there are any significant differences between these plant species. And straight off the bat, you can see that the flax plant had a fairly low alpha index compared to the others. And furthermore, you can see that the bush lawyer, Discaria tomatu, leptospermum, and the Pohutukawa plant had fairly high alpha indexes, and all 4 of those plants were found in the same region in the southern island. And more we're looking at our pairwise significances, we can see that the flax plant and the Kanuka plant were significantly different from the Leptospermum plant and the Pohutukawa plant, which is interesting because the flax and Kanuka were primarily found in the North Island, whereas the other 2 were found in the South Island. And moreover, we can see that the dominant microbial phyla we found were predominantly proteobacteria, actinobacteria, and firmicutes. And this graph right here just shows the relative abundance of those different bacteria phyla amongst all of those plant species. Next, we looked at the biogeography between the North and the South Island. And so again, this is another alpha diversity graph. And on the x-axis, we have the different plant sites. And all of the South Island sites are grouped together in white, and the North Island sites are grouped together in that coral color. And again, we have the Shannon diversity on the y-axis. And so what we found here was that on average, the Southern Island sites had higher alpha indexes than the Northern Island sites. And we conducted another pairwise post-hoc ANOVA test. And something that we noticed, which was significant, was that the Lincoln, New Zealand site, the Silver Peak site, and the Sumner site in the Southern Island were significantly different than the Kapoor's Road site, Lake Rotoiti site, and the Rotopico site. And that's interesting because it shows that there's a trend of difference between the North Island versus South Island, showing that there's on average higher alpha diversity in the Southern Island than the North Island. And lastly, we looked at the beta diversity. And unlike the alpha diversity, which just shows richness and evenness within one community, the beta diversity compares different communities. And in this graph, the points that are closer together represent more similar microbial communities. And as you can see, there's a cluster in the middle where most of the plant species can be found. However, on the x-axis, which accounts for 11.2% of the variance, we see a dissimilarity between the flax plant and the Manuka plant. And this was interesting because when you look back at the map, you can see that the flax and Manuka plant were sampled in similar sites in both the North and South region. However, the fact that they show dissimilarities points to some indication of plant specificity happening here. And that's something that we would definitely want to research further in future works. And so ultimately our main takeaways were that the average alpha index is greater in the southern region compared to the northern region, indicating some geographic patterns. And we also noticed that the flax plant is very different than the Manuka plant from our beta diversity, indicating that there could be some plant specificity. And so in our future work, we would like to examine how regional temperature and local pollinators are affecting these nectar microbial communities. And we hope that all of this will point us towards more information and insights into nectar microbes in New Zealand, especially for these endemic plants that are just being surveyed for the first time. And lastly, I would just like to acknowledge the local iwi for permitting the field sampling on their lands, as well as for everyone of the REU mentors and my mentor, Callie, and the whole team Nectar Micro. Thank you.
Sorry, going the wrong way. Great work there, Saye. Any questions on that presentation? Maybe I'll start. For the difference between the North and the South Island, did you think that that was mostly due to differences in the abiotic conditions like temperature or rainfall, or was it more due to species interactions?
I think it might be more due to temperature because the southern island does have a much lower average temperature than the northern island. But yeah, that definitely would require more studies on that. Given that the beta diversity, there wasn't much more dissimilarities between the plants, I think it points to more abiotic factors.
Thank you. Would any mentees like to ask any questions? Okay. Then thank you, Saye. Last up, we have Brianna and Jonathan. And they are giving a joint presentation. Who would like to control the pointer?
I think Jonathan can control it first, and then after his video, I can control the pointers. That would be okay with you.
Sounds great. You should be able to click to go to the next slide when you're ready, Jonathan.
Okay. I can do it through Zoom?
Yeah, you should just be able to click on the screen.
Click on the Zoom page. Okay.
Yeah.
I see. Oh no.
Too far.
You hear these? Just click there. Oh no.
There you go.
Right here?
You're good. Yeah.
Okay. Hello everybody. My name is Jonathan Barros, and I'm doing a co-presentation with my lab partner here, Brianna Martin Villa, and our mentor is Callie Chappelle, and our primary investigator is Tadashi Fukami. And we did a Fukami Lab summer research simulating the effects of evolution on nectar microbe meta-communities. We hear these words all the time. Bacteria. And I'll play this video. We hear these words all the time. Bacteria, fungi, viruses. But what do they all have in common? And what purpose do they serve in our daily lives? Or more importantly, in our environment? Well, although these microbes are not visible to the naked eye, they play an enormously important role in both our overall health and well-being as an individual, and further, in ecological communities. For instance, the food we eat gets broken down by tiny little microbes in the gut. These are the same microorganisms that keep things flowing, protect us from getting sick, and keep us energized. Collectively, these microbes function in diverse ways across almost all species of life. We're going to explore a particular example involving nectar microbes and use ecological principles to study competition amongst these microbes. Let's take a moment to connect the big picture. I'm sure most of you have heard of Charles Darwin's theory of evolution by natural selection, a process by which organisms change over time as a result of changes in heritable traits. An extension of this theory is that ecology and evolution go hand in hand. That is, most people think of evolution occurring over hundreds of thousands of years, but evolution can also occur over shorter periods of time. Therefore, we should study evolution in light of ecological dynamics, or eco-evolutionary dynamics, which is an approach that focuses on the individual-environment relationship over shorter periods of time. This summer, I've had the opportunity to research with PhD student Callie Chappelle, along with Stanford professor Dr. Tadashi Fukami in the Fukami Lab at Stanford University. I'm also working with Brianna Martin Villa and a group of undergraduate researchers called Team Nectar Microbe. And my name is Jonathan. Our lab studies communities of bacteria and yeast that inhabit the nectar in a species of flower called Diplodocus aurantiacus, or the sticky monkey flower, here in Northern California. We consider a landscape of sticky monkey flower plants to be a meta-community, or a community of communities. The plants themselves make up a group of communities, and individual flowers make up communities within plants. These individual flowers essentially make up individual microbial communities, which are all connected to each other through pollinators, typically hummingbirds and occasionally bees. Recently, the lab has been interested in studying ecoevolutionary dynamics and how they relate to priority effects. Priority effects are when the order of arrival of species to a habitat influences how they grow and interact. And because of priority effects, when bacteria and yeast grow, they compete and usually only one microbe dominates. Priority effects are important because they can help us understand how traits evolve by focusing at the level of metacommunities. We wonder, how do changes in the competitive dynamics between bacteria and yeast influence evolutionary changes in communities of flowers and in communities of plants? Could priority effects between bacteria and yeast create the conditions for evolution to happen rapidly, changing the ecological dynamics and ultimately promote resistance to priority effects? How do we approach answering these questions? Well, we can study ecoevolutionary dynamics in nature, in laboratory settings, or in theory. Studying systems in nature is helpful because we can see it in real time, without artificial tampering, and researchers from the lab have field data to suggest that bacteria and yeast interact competitively because of priority effects. However, it's often hard to isolate this particular ecological feature, since replication and controls are harder to implement. But in a laboratory, we can precisely study ecoevolutionary changes by keeping controlled variables and maintaining close observation. This is how our lab was able to evolve yeast to compete with bacteria in a synthetic nectar environment, and how it was determined that bacteria change the chemical properties of nectar, which consequently impacts the plant-pollinator relationship. Also, to better understand and predict ecological patterns in nature, we can make use of theoretical studies, such as a computer simulation, like the one Brianna and I are currently working on, where we are exploring the myriad of possible scenarios by testing from a set of parameters. And so, it's important to incorporate experiments in each of these settings to have a well-rounded understanding of our study system. We are interested in these research questions because they can be applied beyond the fields of ecology and evolution. For instance, antibiotic-resistant microbes in agriculture, or nurturing healthy microbes in the human gut, and even microbes on the skin from a vaginal delivery. By studying the competition of microbes on a less complicated system, it can help us understand the microbial communities of a more complex system, such as the human microbiome. We can expand our questions and ask, can microbes in the human gut evolve rapidly to resist priority effects, and thereby having grave consequences such as a change in the likelihood of diseases later in life? We can learn more about individual species, species interactions, and communities at large by understanding how these ecological changes influence evolutionary changes. So the next time you make a cheesy dish or drink kombucha, you can say thanks to the ecological and evolutionary processes shaping the microbiomes we love. So here we can transition to handing over control to Brianna. So thank you very much.
All right. Okay. Hi, everyone. I'm Brianna Martin, and I'm a rising sophomore at Stanford University. And this summer, I had the opportunity to work with Jonathan and Callie as our mentor to work on this project, and I'm just going to be continuing to talk about our project. So in our project, we focused on creating a simulation model to address the following research questions. How would changing priority effect strength across the flowering season change the community dynamics in the meta community? And you may be wondering why we chose to take a simulation approach. Well, we wanted to be able to create predictions of what is going on in the field without having to do direct, oh my. It's shocking. Okay, I think we're good. What is going on?
It looks like you got cut off there. Let me try to fix this.
Okay.
One moment, everyone. Is this the right side?
Okay. Yeah, it is. Okay, I think we're good. Do I have control of the screen, Acer?
Yes, you should.
It's not allowing me to move anything.
Okay, one more time. Now? Someone's moving it.
No.
Try clicking.
Yeah, I still can't click.
Okay, well, just tell me whenever you want to move the slide and I'll do it for you. How about that?
Okay, that sounds good. All right, so can you go back to the previous slide, please?
I think I can. There it is.
Wait, I think I have control now.
Okay.
Okay, sounds good. All right, so going back to what I was talking about, so you may be wondering why we chose to take a simulation approach. We wanted to be able to create predictions of what is happening in the field without having to do direct experimentation. So we use an existing simulation model by a former PhD student at Stanford, Ji-Hwan Song, and modified it to include the parameters we were investigating. So now I'm going to explain most of the parameters in the base simulation that Song wrote. And in the base model, there are flowers that get pollinated by hummingbirds and bees. And these pollinators carry microbes, which are put into the flower and will either be colonized by yeast, bacteria, co-dominated, or have no microbes at all. So at the beginning of the flowering season, the flower is not colonized by any microbe because it has not been visited by a pollinator yet. However, when the pollinator comes to get nectar from the flower then gets inoculated, and the flower will be colonized by either bacteria, yeast, or both, and in this case, bacteria. So flowers that have not been colonized yet later get introduced to the community. And as before, a pollinator will come and pollinate the flower. And in this case, because the pollinator visited a flower that's bacteria-based, the new flower that surrounds it has a high probability of becoming bacteria-dominated flowers as well. And these events of pollination are happening across the landscape and are all connected via a meta-community. And the meta-community is many individual communities linked by dispersal. So the original plot that Song wrote shows the relative abundance of colonized and non-colonized flowers in the meta-community throughout a flowering season. And the trend here is that the percentage of flowers in each category stays approximately the same across the flowering season. So you may be wondering, but what about priority effects? Song simulation doesn't take into account that a community that starts with some microbe can change different community states. So the order of arrival of the microbe is extremely important in determining the dominance of the flower. For example, if a flower is yeast dominated first, and then a new pollinator comes carrying bacteria, the likelihood of that flower becoming bacteria dominated is very low. It is more likely that the flower would become co-dominated as opposed as bacteria dominated. So we modified song simulation to build in this change in community state. Here's an example of a flower that starts as yeast dominated, but it could have been bacteria dominated. Over the lifetime of the flower, there's a probability that the flower will transition into another community state. So the first possibility would be that the flower would not change community states. And this is, there is a high probability that the flower will start and end being yeast dominated in this example. There's a 60% chance of this occurring. Another possibility would be that the flower would transition into an entirely new state. So there's a very low probability that a flower would transition from being yeast dominated to entirely bacteria dominated. And in this case, there's a 10% chance. And another possibility is that the flower would become co-dominated. And in this case, there's a higher probability that this would occur, 25%. And the last possibility is that the flower would end up containing no microbes. And there's a very low probability of this occurring. So over the course of the flowering season, we hypothesized that the yeast evolves to resist the effects of bacteria. And this decreases the probability of bacteria becoming dominant and therefore increases the probability of yeast-dominated flowers. So this rapid evolution of the yeast impacts the probability of a flower changing community state. And at the end of the flowering season, the yeast has evolved to not be affected as much by other microbes and as much as priority effects. So therefore, the probability of a flower changing community states would decrease as time goes on. So this represents the meta-community at the beginning of a flowering season before the yeast has evolved to resist priority effects. And at the end of a flowering season, some flowers have transitioned to new community states, some did not change at all, and some died while new flowers were introduced. So we expect to see that the yeast evolves to be more resistant to other microbes, as seen here. So returning back to the original model, we see that the level of the flower abundance stays approximately the same. However, I want to emphasize that this is hypothesized results. We are still working on finishing our code. So in the new model that we expect to see, we expect that because we added the evolution parameter, we will see the different categories all increase, decrease, all change, and not stay the same throughout the flowering season. So for instance, because the yeast has evolved over time, we expect the number of yeast-dominated flowers to go up, and the number of co-dominated flowers to go up as well, and then bacteria-dominated flowers to go down. And overall, we conclude that rapid evolution might dramatically influence what we see in the community, and this theory can be applied to various situations and communities, and the simulation is a great way to help us understand what is happening in the field without having to do the experiment. Here are references, and we would like to acknowledge our amazing mentor, Callie Chappelle, and Stanford professor, Tadashi Fukami, for their support and guidance throughout our research project. And we would also like to recognize the members of the Fukami Lab, as well as a special thank you to the Tea Nectar Microbe for all their help. Is there any questions?
I'm going to take back control from you there, Brianna.
Sounds good.
Any questions for Brianna and Jonathan? I was curious about the interaction between the yeast and the bacteria. Is it all nutrient competition, or are they creating chemicals that are actually interfering with the bacteria? Say, can you have a yeast that's producing an antibacterial? Have you guys, do you know anything
I'm
about
not
that?
entirely sure what you're referring to right now, but
Okay.
the growth of bacteria does have like strong negative effects in the flower. So it changes the
Okay.
chemical properties of the nectar, which then in turn reduces the attractiveness to the pollinators, which is why we expect that the bacteria-dominated flowers will decrease as time goes on, whereas the growth of the yeast-dominated flowers will increase because the pollination rate is like a positive trend.
That makes sense.
And maybe to go off of that a little bit, we did take a community-based approach, and so we were looking at the communities and total population of bacteria as a whole and how they interact with other yeasts, and rather than a individual-based approach where we would most likely look at those makeups that make differences in the total population.
Got it. Thank you. Last chance for any questions.
I have one more question. So starting out your REU with Callie, did you both have experience with bioinformatics? And if not, how did you go about learning those skills virtually?
Good question.
I personally had no prior experience going into our project, and I only had taken a CS like intro class my freshman year, so it was pretty challenging trying to understand everything towards the beginning of our research project. But Callie was a great mentor, and she taught us everything that we know right now. And we also looked at other resources that were really helpful just to like learn R and learn how to create a simulation. And asking a lot of questions was really useful for us.
Yeah, definitely. And we both went in sort of with no experience and started off. Originally, we were going to program in Python, and so we were starting off from scratch and just watching basic videos on what to learn about Python. And then we transitioned to learning about R, and so there was a bit of a learning curve, and we started off just learning everything from scratch, and then we then transitioned to learning more about simulation code. And so then understanding that, we progressed and tried to implement some ideas, and then to address our questions and our project.
All right. Well, I think that is a great segue. I'm gonna close up now and just take this last moment to say congratulations to all of our mentees. This has been a really, really hard setting to do some really great science, and you guys pulled it off. So you should pat yourselves very firmly on the back. Yeah, just appreciate that this is not how science normally works, and a lot of people think that it's impossible to do science this way, but I think you have shown that it is definitely possible, and you can come up with some really impressive results. So with that, I'm going to close the live stream, and not before I say thank you to the mentors without whom this would not have been possible at all. You guys are outstanding. So closing the live stream, don't go anywhere yet though.
And a big thank you to Acer for